My name's Gavin Noble, Process Optimization Chemist at Bachem UK, and I'd like to invite you all this morning to our webinar entitled From Idea to Application, Enabling Better Peptide Drugs. And this is a Belyntic startup story. First, a little bit about Bachem and who we are. We're a leading producer of peptides and oligonucleotides. We recently celebrated our 50-year anniversary, demonstrating our experience and knowledge in the field. Our headquarters are in Switzerland, and as such, we're listed on the Swiss Stock Exchange. And for further information, please visit our website, bachem.com. I'm very happy to introduce today Dr. Robert Zitterbart, co-founder and head of R&D at Belyntic. Robert will discuss his professional background during the talk, so I'll just say that we've been working together for a number of years now, and we've so far had a very, very fruitful collaboration, and yeah, presented several webinars and looking forward to more work going forward. Before we continue, I'd like to give a quick poll just to get an idea of our audience today. So I'd like to ask, did you watch any of our previous Bachem Belyntic joint webinars? And you'll see responses pop up on the screen. Yeah, we can see we've got a few people who've watched before, but also we've got quite a new audience today, so that's really exciting. And hopefully we'll be able to show you and demonstrate this exciting technology. First of all, though, I'd like to just introduce Bachem. Our business model ensures that we have coverage of all aspects of peptide production, from initial research stage right through to commercialized API. And having a high focus on innovation allows us to transition smoothly through each step of the production, so we ensure that we're the perfect partner right through. Bachem operates out of the Swiss site in Bubendorf. That's our headquarters, but we also have several sites globally, including GMP manufacturing sites in Vienna, Switzerland, and 2 sites in California in the US. We have our Center of Excellence for Peptide Manufacturing in St. Helens, UK, where I'm happy to be based. Here we focus on custom synthesis and synthesis of diagnostic peptides, and is the site where some of the work that Robert will present was undertaken. In addition to this, we also have an office in Tokyo to help service our Asian partners. So why Belyntic and Bachem? Well, Belyntic's a chemistry for healthcare company focusing on peptide drugs, and therefore a solution provider. We are the leading experts in development of large-scale production of peptide APIs, so it made sense to join together. We looked to form a sustainable partnership right from the beginning, and as I mentioned previously, the first evaluation took place in our UK site in St. Helens. Current focus of the collaboration is looking at establishing Peptide Easy Clean for GMP manufacturing at Bachem. As such, we're very proud to host Belyntic again for our webinar today. And Robert will present the journey of Belyntic from an idea between 3 friends to a dynamic and innovative startup company. So last point before we get started with the webinar is at any point during the talk, you may ask a question, and hopefully we'll get time to answer all the questions in the Q&A section. So please feel free to ask. And all attendees' microphones and webcams will be switched off, so don't worry about that. So without further ado, I will hand over to Robert.
So thank you everybody for attending this webinar, and a big thank you for Bachem to invite me to talk about our Belyntic startup story. And also Gavin, thank you for being such a wonderful partner for the past years, as you said. So it was always really fun to discuss chemistry and peptide science with you. And thank you for the introduction. And yeah, and welcome to my talk, a startup story about Belyntic from idea to application. And when we talk about application of our technology, we mainly mean to enable better peptide drugs. Let's switch to the next slide. Let's see if this works. Sorry, I was a bit fast. So, yeah, so in this picture, the agenda of my talk is basically covered. So I will take you from the invention of the technology, starting even before 2016, but mainly taking up speed at 2016, and then I will also talk about the foundation of the Belyntic GmbH in Germany. And now we are in a growth phase where we have a bigger team now and want to tackle the next steps of our company history. But let's go back to the beginning. So the very beginning was at the end of 2005. I started to study chemistry together with these two friends here. So this is an old picture of Dominik Sama and Oliver Reimann and myself. So as you notice, back in the days, I still had hair on my head. And then we started right from the beginning to be friends and just studied together and practiced for exams. And also I found, when I started to study chemistry, I found that I really loved chemistry and especially organic chemistry. At the end of my studies, I went to California to the UC Berkeley and did my master's thesis with Peter K. Vollhardt, also in organic chemistry, but also using a little bit of inorganic chemistry, what was also interesting for the synthesis of morphine, actually. But then I went back to Berlin, back to the Humboldt University, to join the group of Professor Oliver Seitz. He's a true expert in peptides and DNA synthesis, so oligonucleotide synthesis. And there I started my PhD in the early 2011. And so this was a long period, my PhD time. And I took over a project which was using a technique called self-purifying peptide thioester synthesis. And this technique is very useful for synthesizing peptide thioesters. On the next slide, I will explain you what peptide thioesters are good for and what I used it for. And don't be scared here by the scheme on the right side. I won't go in detail. If you are interested in the chemical details, please refer to the papers that are stated here, or also papers that I have published describing this technique in detail. I just want to point out one thing, that this HPLC, it was an HPLC-free purification of peptides, peptide thioesters, and it utilized the synthetic resin as means to purify the peptide. And this was possible by using a macrocyclic peptide structure here. Okay, and this technique was invented by Oliver Seitz, so my doctor father, and was developed by Franziska Mende. She's now called Franziska Thomas and is now a professor in Heidelberg. As I said, in 2011, I started my PhD using this technique, and it was good in a way because I found out it's very useful to synthesize many peptides in parallel and pre-purify them. And but the downside of this technique was it was only applicable for thioesters. And also what I found out, that truncations that should be removed by this technique weren't always removed. So they were sticking to the synthetic resin. So keep this in mind. What I did with those peptide thioesters is shown here on this slide. So I used those peptide thioesters here in a reaction called native chemical ligation. This is a very useful reaction where you can join peptide thioesters with N-terminal cysteine peptides to to create a native chemical, a native peptide bond. And you can do this with unprotected peptides in a very selective and nice fashion. And I did this in a way that I used these N-terminal cysteine peptides as a peptide hydroxides. I first immobilized them via hydrazone formation on multi-titer plate surface here, so in a 96-well format. And then later on, I added those self-purified peptide tire esters to it. And the aim was to synthesize protein domains to analyze those in binding assays. And I analyzed phosphorylation patterns. If you are interested in this, also please read my publication here. But I just wanted to show this to you to explain native chemical ligation, and also what I found here, because I was struggling with immobilization techniques for peptides, and I found that these hydrazone formation here works very robust, and therefore I thought this is good technique to use. So please also keep this in mind. But now we go a step back to the three friends. So at what we thought at this time, at the end of our PhDs, so in the end of 2014, we met together with some beers, and we were just thinking about, hey, what should we do at the end of our PhDs? And we said, hey, why don't we start a company together? Maybe crazy idea, but would be fun to work together like in the beginning of our studies. And then we said, yeah, why not? Let's think about it. And we put all the ideas of our PhDs, also innovations we were working on the table, and said, so what of those ideas are worthwhile to proceed? Oliver, for example, was working on antibody drug conjugates. I was working on the self-purifying peptide ester synthesis. Dominic was working more on the material side on bead-based assays. So after some time, we said, okay, we are not decided yet what idea we will proceed, but at least the decision to at least consider starting a company together is appealing. So what we did, and this was really nice, we joined a workshop at the Free University Berlin, because Oliver at this time was doing his PhD at the Free University. So we were allowed to, all three of us, attend this workshop, and it was very good, called ProFund workshop. And at this workshop, we just further developed the idea, and we're thinking about what ideas of us would be worthwhile proceeding. And also we got some feedback from the coaches there and from the other students that actually what we do there is interesting. And then we analyzed the ideas and found that actually my idea, so the self-purifying peptide ester synthesis, has the most potential to be proceeded. But as I said, the self-purifying peptide esters technique was not ideal yet. So we were also thinking about what to improve. And fortunately, also my mentor, my professor, my PI, sorry, Oliver Seitz, also agreed that I can further investigate this idea. And what was the idea from the beginning? It's shown here in a very old scheme from this time. So we were thinking about tagging target peptides with the catch molecule or linker, and also we wanted to apply a filter material that should capture this target peptide, but not the rest we wanted to remove. So this is the first linker generation I created in my PhD time. And as you notice, it's again a hydrazone formation to capture the peptide. The next step was to wash out the truncations, to remove the truncations and synthetic dirt. And the third step is a chemical stimulus of the linker to release a purified peptide. And this was done by basic stimulus here. At the first linker generation was not ideal because it released a divinyl sulfone here, but it was the easiest to synthesize. So now we had the basic idea, but still we were really shy about starting a company together and also about our idea. In fact, when I told chemists at the Humboldt University about my idea, they were not so positive actually. So they said, do you really think this is going to work? And aren't big players in the field already having those techniques? And but again, in this workshop, the atmosphere was really encouraging. And one coach there said, okay, you now learned a business model canvas. It's a technique where you put in a different business model and you just see what's working and what's not. And so he taught us there in the seminar, and he said to us, okay, there is this business plan competition called BPW. It stands for Business Plan Wettbewerb, so competition. And it's one of the most renowned business plan competitions in Germany. And he said, so this coach said, yeah, just apply there. You learned the principles, write down what you learned, apply there, and you will see what you get. And so we did it, and we never thought about any positive response, but we were invited to the jury and pitched there. It was very exciting. And then it was even more exciting that we even won the first place there. So, and this was really the starting point to be more to be more concrete about this, because we thought, okay, like business experts said, this is an idea worth following. So we said now we take it serious. And so at this time we also incorporated the name EnviroPep, and also we went to the Humboldt University, to the founding agency there, because it was at the beginning not so easy for us, because so I was from the Humboldt University. Oliver started his PhD at the Free University, and then his professor joined the Leibniz Institute, and Dominic was located at the BAM. I will explain you in the next slide what the BAM is. It's a federal institute. And I was from the Humboldt University, but since we used the idea from the Humboldt University, we went to the Humboldt University and the founding center there, and they were also very helpful and very nice, and they told us about some program called Exist Forschungstransfer. And this is a very nice project in program in Germany, where you get funding for especially ideas that transfer ideas from academia to the business side. So this was very well suited for us. So we applied there and were even invited to the jury commission, but unfortunately at the jury we got a negative votum. The main reason was that they said you don't have a patent yet. And thus, since we already proved that the technology worked, we filed our first patent, or the Humboldt University filed the first patent in January 2016. And then at the end of January 2016, we could apply again for this Forschungstransfer. Second try, we were again invited to the jury commission, and now they granted us this Exist Forschungstransfer. So we were very happy about it. This meant we could start with our dream. So this was a very happy moment. But the next problem arose, okay, but we need a lab for that. The Humboldt University didn't have free lab space at that time. And so we were really fortunate that the BAM, so the Federal Institute for Material Testing and Research, also here in Berlin, Adlershof, where Dominic did his PhD thesis, was offering us lab space. So this was really nice. Also, due to the Exist program, they asked us to include a team member with business expertise. So we found Andreas Regneri, and he became the fourth founder of Belyntic. and complemented the team with business expertise. So, and then we entered this exist phase here. And our earliest contact with Bachem was actually after we have won this BPW, the business plan competition. There was a press release, and Günther Leudel saw this press release and called us. And he wanted to investigate the technology already. At this time, we said, okay, it's very early. Please wait a bit till we have our own lab and such. And we had that. So therefore, we started this collaboration with Bachem together. Also, Gavin was involved there. So we further developed this linker principle we had to a more sophisticated linker, but still base label. So again, the chemical details are described in this paper here. Here, first we investigated the hydrazone formation, as I explained at the beginning, and oxime formation. So when you have an oxygen atom here, and found that the oxime formation is actually beneficial, for example. We also investigate several base label linkers, and we found that the basic principle works very well to get a purified peptide without truncations inside, high yielding, and high purities. So this was a good start. But in the evaluation of the base label linker system, also together with Gavin, thank you again, he synthesized many peptides for us that we under industry standards that we investigated then for the purification. And together with Gavin and Bachem UK, and also in our lab, we found, unfortunately, that these base label linkers underwent base-induced side reactions. So for example, when we used N-terminal threonine or serine sequences, the hydroxide attacked the carbamate structure of the linker, and oxazolidinone moieties were formed. Also, aspartamide formation was an issue for certain sequences. And the most profound impurity we found was on certain sequences with an arginine glutamic acid. Apparently, the basic conditions were producing an citrulline formation from arginine. So as a conclusion, we found that this principle does work and it's a nice linker system, but it's not generally applicable. And we wanted to have a generally applicable linker that works for all peptides. Therefore, we invented a reductively cleavable linker system because we figured that reductive conditions for linker cleavage should not harm any peptide. Therefore, we invented this linker structure, and this was also evaluated by Bachem UK. And at first, it looked very promising. At this time, also in May 2018, the Belyntic GmbH company was founded under the name Belyntic. As I said previously, we were known as EnviroPep. So now we were really happy, and we thought, yeah, we are founded now. We have a generally applicable linker system. Now everything can start. So we went to Bachem UK and had a good time with Gavin. He was a really kind host there, and we tested the linker system, and everything was nice except that the chemistry didn't work as planned. So in contrast to our tests in our lab, the yields were less in the Bachem lab, and we traced it down to the acid lability of the linker system by TFA. So to investigate this further and to really put better numbers on that, we created a test system using our linker molecules and propylamine to generate a surrogate of a peptide. So to have like a linker propylamine species that we can investigate in solution under a TFA treatment. So what we did was we generated this propyl amine carbamate, linker carbamate, and then we treated it with different TFA cocktails. And this is shown also here at the bottom. So for example, if you use a very commonly used cleavage cocktail, reagent K, where you have tiols inside, it's actually a nasty cocktail, but it gives usually good results. So if you use this one, you have a high decomposition of this propyl amine linker species. If you use TFA TIS water cocktails, then the decomposition is less. But overall, the loss of linker modification on peptide would account for 60 to 90% loss of this linker modification within 2 hours. And this was really unacceptable. So therefore, we stopped selling the linker because also other potential customers reported that their yields weren't so good using our linker. But without any product to sell, we needed money. Therefore, fortunately, we got financiation by the investment bank in Berlin to further develop our linker molecules. And we developed many of those, but some of the new linker types were showing halogenation here on this position. So mainly what in terms of synthesis, the highest yielding and cleanest products were by getting this monobromo and dibromo brominated linker species. And we did the same tests as I described before with those. And when there was one bromine or two bromines attached, there was no decomposition during TFA treatment. Also, we got more financiation by the high-tech Gründerfonds. So we were also very happy about that. This brought us over this kind of troublesome time where we didn't have any product to sell. And from all the linkers synthesized and tested, the best one turned out to be the monobrominated linker here. This shows efficient stability against TFA and also has additional benefits. For example, that we were able to use DTT as a reducing agent instead of triphenylphosphine as we did before, and also enables even disulfide modification, as I have shown in a previous webinar. So I know that the majority of you didn't attend this webinar so far, but they are all available on YouTube, so you can watch them there. But you can also have a look in this publication if you want. So also another issue was that potentially those linkers could have been explosives since they have an A site. Also, the amino oxy moiety here could be a potential explosive hazard. Therefore, if you create a new chemical entity, but not in pharmaceutical terms, but as a bulk material, you have to classify it at the beginning as an explosive, and you have to test if it's an explosive or not to get out of that. Fortunately, the federal institute that does this in Germany is the BAM, where we are located at, and they did this testing, and they found out, fortunately, the linker is not explosive and it's not toxic. So even more a reason to go out now to sell the linker molecule. So we created with this a research kit where you have everything inside, pre-packed cartridges where the filter material is in, and the linker molecule and all other chemicals you need for the process. Also a developer kit that is more a bulk kind of material. So it has the filter material that is agarose, aldehyde modified agarose, and the linker molecules that you can use for for high numbers or large scales. What we were also very interested in to use our technique for high throughput purification of peptides, because one big advantage of our technology in contrast to HPLC purification, so high pressure liquid chromatographic purification of peptides, is that it can be run in parallel and that only little negative or positive pressure is needed to process. And you can do this by liquid handling. So whereas for HPLC purification of, let's say, 96 peptides, you have to run it 96 times over an HPLC. You have to think about gradients and you have to think about thresholds and how to collect your samples. With our Peptide Easy Clean technology, PEC, you don't have to think about it. You just treat each peptide equally and you can run it parallel. So we knew that a parallel purification, especially in 96 well format, would be appealing. But the question is how to do it. So this was early attempts where we just used a deep well plate, a filter plate at the top, and something we found in a lab. And it didn't work very well, but we were fortunate that Stefan Lüttke joined our team during his master thesis, and he later on also joined to work for Belyntic. And he, together with me, transformed this basic idea into a really a product where we have now special filter plates with the PEC material inside, and we have a high throughput kit that comes with everything you need. We are also selling those vacuum chambers for manual processing of 96 peptides or 48 peptides in parallel. Also, I will, in a few minutes, I will talk about automation in this regard. So, and we were planning to release this product in April 2020. So at this point, we thought, now we are really ready to take off. We have a linker that's generally applicable, that works well. We have 3 really nice kits to market on the peptide market, and we have really USP, especially in the high throughput kit. So, and then everything turned out to be a little bit different. So yeah, first of all, sorry, this is the benefits again of the linker system. So it's a first-in-class parallel manufacturing. It can be used for peptide modification, as I've shown already in a previous webinar that is also available on YouTube. Also, that it's something I didn't mention before, but this was also one big motivation, that the linker molecules enables significant reduction in solvent consumption compared to HPLC. And also for large-scale peptide production, it shows an increased productivity at larger scales. So, and with this, all these benefits in hand and the products in hand, we really thought at the end of 2019, we can really take off now as a company. But as you know, then a little problem occurred. So everything then changed with a Corona crisis approaching. And what this meant to us is shown on the next slide. So due to our funding structure, we had some obligations to employ people. So our staff was just growing at the beginning of 2020 to 8 people. And this was then due to the Corona pandemic, the time when the sales activities became very difficult. So, so what that meant was that virtual conferences, for example, weren't helping, because for convincing people to apply a new job, to use a new technology, you have to rely on personal bonds, and this is very difficult to establish in a virtual conference, for example. Also, already planned projects with customers and potential customers were put on hold because they didn't know what the next months will bring. Also, already planned evaluation of our technologies all couldn't be run due to lab staff being in home office, or due to the fact that we were planning to go there and that we couldn't due to travel restrictions. And as a summary, basically, also nobody wanted to take any risk anymore with using a new technology. So everybody was relying on the status quo because everything was uncertain. On the other hand, there were also opportunities in this time. So, for example, there was a high demand for peptide libraries to, especially of the coronavirus epitopes. And since we did just release this high throughput kit, we said, okay, if customers don't want to use our technology, we use our technology ourselves to provide peptide libraries. And this worked very nice. Also, we helped with those peptide libraries in the fight against COVID-19 by understanding the virus. And we were also very happy that the spec-purified peptides behaved very well in all biological assays of our customers. So they were usually compared to HPLC purified peptides in 95% purity and behaved equally well or even better. So therefore, we provided peptide libraries, first of all, to improve our PEG technology, to learn about the generally applicability of the PEG technology, because we now purified ourselves over 2,000 peptides now. So we know what peptides work very well and what peptides are troublesome, and we can improve on those and find out what is the reason that some may not work as well, and when we can incorporate means to prevent those peptides from not working so well. So we could compensate for revenue losses in our original business model, but also very interesting. Since we entered this field of peptide libraries, we got in contact with clinics and also academic researchers that were investigating COVID. And together with Professor Natalio Gabi from the University of Bonn, we also started to start an own vaccination program against COVID-19. So we got a SIM funding for that in late last year. And we could also even further improve on that by partnering with Professor Leif Erik Sander. He's the #1 infectiologist in Germany. He has recurring media presence because he is very renowned in the field of vaccine development and application, also in the COVID context. And together with him and the Charité, we further expanded our vaccination platform to also have a general applicable vaccination platform against viral diseases. And for that, we were counted with a funding of the Sprint challenge. It's the money comes from the Bundesagentur für Sprunginnovation. And to summarize what we do in this whole vaccination project, that I cannot tell any details about it, unfortunately, right now. But the basic idea is that Belyntic creates a best-in-class platform for rapid and safe intervention against viral disease outbreaks to provide life-saving therapeutic vaccines to patients. But maybe you think now, okay, now Belyntic turns into a biotech company. This is not entirely true, because we can only do this because we are an expert in peptide manufacturing and also have the good partners like Bachem, where we could potentially transfer a project to GMP manufacturing. And our Peptide Easy Clean technology enables to synthesize more complex peptide molecules than it's being possible with HPLC. So also by following this biotech route, we are also improving our technology. But more concretely, we're also maintaining our core business, also together with Bachem, where, as Gavin already mentioned, the GMP applicability of manufacturing of peptides using our technology is constantly investigated, and it was looking very good. So the PEC technology is GMP compliant. And also, as mentioned earlier shortly, is we are also working on automation of our technology. So what's currently available is only a manual process, but we cannot disclose how it exactly works right now, but it works with 2 machines, or at least 2 machines in a 48 and 96 well format in a high throughput fashion to purify peptides in 10 to 25 micromole scale. Also, a larger scale processing is possible with 6 to 24 cartridges, so 6 to 24 peptides, and scales of 50 to 200 micromole, potentially even 1 millimole scale. Also, one thing we can disclose is the work together with SpinChem to investigate this rotating bed reactors that also work very nice together with the PEC technology, and is one possibility to scale up the PEC technology into kilogram scale, actually. So this brings me to the summary slide. So I want to just summarize my talk and also tell you about the lessons that I learned. So one thing we were told at the very beginning is that 3 things are very important when you think about starting a company. So these are shown here in this graphic on the right side. So what's very important if you want to start an innovative startup is the team. You should have a business model, and this is obviously based on your innovation. And we were told one of the 3 elements don't have to be ideal, and you should be flexible there. But from my understanding, it's the team should be not flexible because the team will be always there, and you should rather be flexible in the business model, or maybe even in the innovation you have. So lessons I learned was for sure, follow your ideas. If you think yourself that the idea is worthwhile proceeding, and you see a market there and you see a potential there, don't rely on others that tell you that this won't be a success. It may take a time, but if you think it's worthwhile proceeding, it is worthwhile proceeding. As I said, be flexible in the business model. As we did in the Corona crisis, we were going away a little bit from the kit model, from the kit business model towards ourselves providing peptide libraries. And a good team is most important, because a good team will help you through difficult times. Also very important, seek industry feedback as fast as possible, as we did with Bachem, because when you talk to an industry insider, they will tell you something you cannot predict or foresee. So you should always be bold and talk to industry experts and test always your hypothesis. And if they turn out to be wrong, then adjust accordingly. So thank you very much for your kind attention, and I'm also very happy to answer any questions in the Q&A session.
Thank you very much, Robert. It's a really fascinating insight into your company and the history there. It was really impressive as you managing to keep your momentum going and seeing opportunity from all the challenges you faced. Yeah, so that's really great.
Thank you.
So, yeah, quick poll for our audience. After seeing our talk today, do you see a role for catch and release purification in your manufacturing processes? So yes, you're already using it, or yes, you're planning to use, or not at present. See the answers popping up now. It's quite a mixed spread, actually. A couple of people already using. Yes, planning to use. That's a very good result for you, Robert. And a few people yet to be convinced, but yeah, very positive. Okay, so we had a couple of questions during the session. Oh, excuse me. So one question that came through, Robert, have you investigated the ARG to citrulline conversion under basic conditions in any more detail? And do you have any publications about this side reaction or any other knowledge about it?
So in this peptide science paper that I had on the screen, there we also cite a literature, a reference that was describing the same observation. What was a bit surprising to us was that from their description they used very harsh conditions to transform the arginine into citrulline. So what they used, I'm not sure about the exact pH, but it was 13 or 14. So as basic as you can go with barium hydroxide.
Mm-hmm.
And it seemed that this very sequence we had was especially prone to this transformation. And we had like 50% citrulline afterwards. But we didn't further investigate. So then it may be that even if you have this same like dimeric sequence, so the arginine and glutamic acid in a different peptide, that you don't get as much citrulline formation as in this peptide, but you will get it to some extent, I guess.
Okay, thank you for that.
But yeah, sorry, if you are interested in the paper, in this reference, whether look in the peptide science paper, or if you don't find it, just write me an email and I can send it to you.
Okay, thank you. Another question, and what were the most challenging parts in growing Belyntic? And further to that, what future growth plans do you have?
Yes, so most challenging was certainly the period when we were just founded, and we, a little bit naive, I would say, was thinking, okay, everything is set. Now we just start to sell the kits, and everything will be fine. So I think this is a common mistake for especially science startups, because they always think about just about their technology, and they think as soon as the technology is ready, everybody will be so fascinated about their technology that everybody will just buy it right away. So this is one constant struggle I think everybody faces with innovation, that you have to convince people that your technology has a benefit, and they have to change their behaviors towards it. So this is always difficult. And for us, it was especially difficult because we didn't even have the technology ready when we thought we had it ready. What was revealed basically when we investigated the technology at St. Helens with you, Gavin. So this was basically the most challenging time. So we didn't have money at this time, so we cut salaries, and and had to pay the rent, and so this was tough. But as I stated, the Corona crisis also wasn't that easy. But fortunately enough, this library demand was so high that we very quick could cope with this revenue loss.
Okay, great. And future directions? Where do you see the company going?
Yes, sorry, there were two questions. And as I stated in the last slide, so we are now entering the vaccine development field. So like some of us will will be mainly occupied by this part, so moving towards a biotech kind of direction, and the other part will remain focused on the core technology and further development of the technology. So there will be growth, especially in this biotech part, because we got now money for that. We are also seeking more financiation for that. So there will be big growth there. And depending on what more project to come for the core technology, there will be hopefully also growth. One thing I want to mention, we will move mid of next year into a bigger lab to also have space for new people. So this will be exciting.
Great growth going forward. Another question from our audience says, does the patent impact the willingness of customers to investigate your technology?
Yeah, so I would say it's very important because, as I said, for customers or potential customers, it's especially in the peptide field, especially when it's going towards API production, people are very conservative because, I mean, they have to maintain their production routes, and they have to investigate every little impurity. So they think very well before they change anything. And they also want to make sure that if they change, that it is long lasting. And one part of this is, if they go down this route and they invest to implement this new technology, also they want to make sure that it's protected somehow. So this is one benefit. They see then, okay, I have this IP protection then around this manufacturing route. So it's a plus. It's for them, but it's most important for us, obviously, and most important for potential investors. So for the customers, it's most important the reliability, I would say, and the patent is a part of this.
Yep, makes sense. Time for one last quick question. It says, could you please explain the effectiveness of your peptide purification of different lengths of peptide, having maybe 1 to 2 miscoupled amino acids?
So yes, when you use our technology, you have to also think this is also one obstacle we have. The peptide synthesis is directly connected to the outcome of the PEG purification. So for example, you should make the couplings in a way, and also FMOC protection, that you won't have missed couplings or deletion sequences. Usually, if you use rather standard conditions, this is not a big problem. So actually our technology does very well what it is designed for, and removing truncations. And to our knowledge, the truncations, and to our tests was, as I said, over or almost 2,000 or around 2,000 peptides purified so far. Really, the main impurities are truncations for an unoptimized synthesis. Okay, when you talk about API production, you optimize a lot to remove those truncations, but especially in parallel manufacturing, you don't have time for that. So you don't optimize and you just run, and there the majority of the impurities are truncations, and capped truncations if you cap well. If you want to have a look, there is also stated in the first publication a capping study. The question was also about length. So I think, and we have proved that with further length of peptides, our technology gets even more superior towards HPLC, because the peptide, the deletion or the truncation sequence get more and more equal to the full length peptide, and therefore it gets more and more difficult to separate those by physical chemical properties like HPLC, and therefore if you have longer peptides, the benefits are more profound using our technology. We have a case study on our website where you can see this, where we purified 84 mer. And this is really it's a triangle, basically before purification. So there are lots of truncations left and right and underneath. and then you really selectively pull it out by pack, and then this would be very difficult with HPLC.
Okay, thank you very much. That brings us to the end of the Q&A session and the end of the talk. Just a reminder, if you have any more questions, you can always contact us via the email webinar@bachem.com, or contact Robert directly via belyntic.com as well. And make sure to check out our socials as well. And again, thank you again, everybody. Goodbye.